Արփայի անդամներում, որ ինձ հրավիրեցիք, մեր կորձերի մասին խոսենք, ցեզի իմ լաբորատորიის կորձերում եւ տարբեր նյութերի մասին խոսենք այսօր եթե պատմեմ ցեզի եւ կառաջարկեմ, որ ինձ հարցում հարցնեք, խոսելու արդեն lecture-ին կսպասելու մինչև վերջը, որ question and answer-ը լա, ինձ հարցում հարցնեք, որ անմատ հարցնա եւ անմատ նույն էջին վրան լա։ Ներողամիտ կլակ, որ մնացած աստվածությունը անգլերենով պիտի խոսեմ, որովհետև ինձ համար ավելի թյուրին է գիտնական բառերը արտահայտել անգլերենով։ And so I'll tell you about the work that we do. I'm a trained microbiologist. I received my PhD at UCLA in microbiology and immunology, and I've been studying microbes ever since. And when most people talk about microbes, my uncle and I were just talking about, my aunt is not feeling well. When we talk about microbes, we always think of microbes or bacteria as being creatures that cause disease, but hopefully I'll tell you today that in fact the great majority of microbes don't cause disease. In fact, they're quite beneficial. And if we can understand the benefits of these organisms, maybe we can develop novel therapies for various different disorders. And so I want to introduce you to a concept that you might already be familiar with, is that we are all covered with bacteria on all of our body surfaces. And so whether it's the skin, and so it's hard to read, but I'll walk you through it. Whether it's the intestines, the mouth, the airways, the skin, or in women, the vaginal tract, there are hundreds, if not thousands, of different microbial species that are living inside or on all of us today. We get these organisms from birth, and I'll describe that process in a minute. And so just to put this into perspective, there are 100 trillion bacteria in each human being. And so if you count the number of bacteria that live inside of you and the number of human cells that live inside of you, there are 10 times more bacteria than there are human cells. And so we are 90% bacteria and only 10% human. And if you think about it that way, this tells you about how rich our interaction is with microbes. We live in a world dominated by microbes. And this is why I love bacteria so much, is because they're a part of our biology. And so this is the number I just told you. And then in terms of the number of different species or different types of organisms, there are at least 10,000 different species. So of this 100 trillion cells, there are at least 10,000 different species that live in all people. And each of us have a subset or some amount of those, usually between 500 and 1,000. So 10,000 in the entire human population, and each of us are different in our microbial composition. And so as far as we know today, and there's a lot of research going on, the process by which we acquire microbes starts at birth. So during birth, birthing through the vaginal canal, we are exposed to microbes. So the fetus is sterile, right, in utero. But that day that we are born, we begin a process by which we assemble all the microbes that we will carry through our adulthood. And so this slide just depicts this process of, in a politically correct way, of acquiring microbes at birth, but clearly through human contact, through our environment, through foods, we acquire all the microbes that we have, and by the age of 3 years old, an infant or a child has an adult microbiome. A microbiome is a collection of microbes that live inside of us. So at 3 years old, we are adults as far as our microbes are concerned. Then the rest of our body keeps growing. And so we're...
With C-section, we still have bacteria?
Yes, that's a very good question. There are many studies that have looked at the difference in microbes in children born through C-section versus natural birth. And the microbes are very different. The microbes that are found in the intestine of a child born through vaginal birth look like an adult intestinal microbiome, intestinal consortium bacteria. The microbes that live inside of the gut of children born through C-section look like the skin microbiome, which makes sense if you think about how children born with cesarean section may look. And in fact, those children born with cesarean section are much more prone to allergic and autoimmune diseases, and I'll describe some of that information. So it's not just that we're different if we're born through C-section, but it has consequences on health. But it's a very important question, and once again, a large area of research. And so, and along those lines of thinking about health and disease, one of the ways that we study these organisms, there are many ways of obviously studying microbes or any organism, but the way we study these organisms is we use models of disease, and we use predominantly mouse models. We've done a little bit of work in rats, nothing in humans yet, and I'll describe our path towards doing human trials. But we use the mouse because it is the workhorse in the biological research community. Mice breed fast. We have a lot of genetic tools to manipulate animals, so that they're a good test paradigm for us. And what we do as an approach in the laboratory is to take disease models and to try and understand how microbes interact with the host, and once again, in this case, a mouse, through the disease process. And when we began this work about 15 years ago, we made the assumption, or I made the assumption, that when we think about microbes, we think about the immune system or the immune response to microbes. So I made the assumption that if microbes were going to have beneficial effects, then their benefits would be on the immune system. And so we started working with models of inflammatory bowel disease. So inflammatory bowel disease is a disease that afflicts many Americans. 1.5 million people in the United States have inflammatory bowel disease. Estimated 10 million people worldwide have inflammatory bowel disease. There are two types of IBD, Crohn's disease, ulcerative colitis. I won't go into the details of the pathophysiology, the type of disease, but these are essentially diseases of the intestines, where the intestine, the immune system becomes activated. The immune system believes that there's an infection. It mounts an inflammatory response. And what happens when this inflammatory response goes on for many, many years is that it causes tissue damage and ultimately the symptoms of inflammatory bowel disease, which are my father just walked into the room, and so I'll stop to say hello. And so inflammatory bowel disease results from this tissue damage, and tissue damage leads to the symptoms, which are severe weight loss, and I'll demonstrate that in animals, but also in inflammation in the intestine as well as bloody stool. And the reason why we chose this model system is because for decades people had shown that there was a microbial component, or believed microbial components, to inflammatory bowel disease. And so here's the evidence that in some cases treatment with antibiotics provided some benefits, so that tells you that microbes might be involved. But there was no definitive pathogen. So inflammatory bowel disease is not an infectious disease. It's not communicable. It doesn't transmit from one person to the other. Right? And for many years, people had looked for pathogens, but my laboratory discovered several years ago that IBD is not caused by a pathogen, but instead by microbes in the intestine. And essentially what we showed is that, as I mentioned, all of us have microbes in our intestine, many, many trillions of different cells. But patients with inflammatory bowel disease, they can't distinguish whether or not those bacteria are good or bad. They think that the bacteria, their immune system believes the bacteria is an infection and keeps trying to mount an inflammatory response. Just like you would if you get a cut on your hand. You get an immune response in that site because there's a pathogen or infectious agent. Patients with inflammatory bowel disease, they go through this chronic inflammatory process, and once again, that's what causes the tissue damage. And ultimately what this leads to is a change in the microbial profile. And what I mean by that is I told you that there are several hundred different bacteria in our intestines. And those bacteria are found in different ratios. And there's a healthy ratio, there's a healthy profile, and then there's an unhealthy profile. And in fact, this is what one of the major findings from our laboratory is that not only is IBD and many other diseases not caused by infections, as they were previously thought, but they're caused by a change in the normal constituents or the normal ratios of these organisms. Okay.
Lately it's become quite a fad to suggest probiotics, which actually are supposed to change the bacterial content in your intestines. Is there any comment you want to make on it, or you want to speak about it later?
Yeah, so Jose brings up the notion of probiotics that you get in health food stores. And in some cases, the spurious or the frivolous claims that are made by these probiotics. I'll touch on this at the end, and I think the reason why I won't pontificate, or I won't drag on this discussion, is because I think after you see the data, you'll see the difference between the organisms we study and the organisms that are commercially available. And so if you think about, once again, the profile of microbes in the intestine, there's multiple ways of looking at this, and I can show you very complex data sets, but I think that the point is made very simply with this slide. So this says ileum, which is the upper part of the small intestine, and then this says rectum, and then in fact even feces. And so when you look across longitudinally across the intestine of a healthy human, you see two major types of bacteria. So I told you there are hundreds of different species, but these species fall into different classes. And there are two major classes. Think of it as, let's say, a Ford and a Chevrolet. They're both different cars, but within a Ford there are SUVs and pickup trucks and sedans, so on and so forth. That's what we're seeing here. So let's say this is this phylum of bacteria and this phylum, one called Bacteroides and one called Firmicutes, are the major types of organisms, major classes of organisms. And as you can see, these dominate in the healthy gut. But when you look in the intestine of a person with inflammatory bowel disease, the profile looks quite different, right? Is that these two major classes of bacteria are reduced, and other organisms have now increased. So you see the yellow are organisms called proteobacteria. They're found in very small numbers in healthy people, but you see that their numbers have grown in inflammatory bowel disease patients. And so what we've suggested over the years is that these diseases, as I mentioned, are caused by changes in the balance of organisms that were there the entire time. Once again, different than, let's say, if you catch a flu or strep throat, an organism that comes from the environment. These diseases are not caused by new organisms. They're caused by changes in the organisms that were there the entire time. And so you can imagine that there are certain organisms which promote disease, and we call these organisms pathobionts, such as pathogens, but pathobionts meaning pathogenic organisms that live inside of the host, or symbionts, which are beneficial. So if a harmful organism increases in number, that can cause disease, or if a beneficial organism decreases in number, that can cause disease as well. Right? And so this is the approach that we've taken, and we've predominantly focused on trying to identify beneficial organisms, those organisms which prevent or can even reverse disease.
What benefits the pathogens to stimulate it?
Why are these organisms even pathogenic? Why are they pathogenic? What's the benefit to the organism itself? Is that the question?
No. How the balance changes with saprophytes and pathogens?
Yes.
What's the reasons?
Yeah, there are many reasons. The genetics of the individual is very important in changing that. Certain antibiotic use, but the major driver of changes in the microbiome is diet. Remember, diet, the food that we eat, right? Because if you think about this, what are the first organisms that eat our food? It's not us. When you ingest food, our bacteria digest it before we digest it. In fact, they help us digest it. Without them, we couldn't extract the nutrients from our food, right? So depending on the food that people eat, it'll change their microbiome. And this is very, very well cataloged, both in humans and in animals. And so, with this notion of trying to identify beneficial organisms, many years ago, and this was work published, it's hard to see, but this was work published in 2005, so the research actually began in 2001. We identified one particular organism that had very unique properties in that it activated the immune system in a way that no other bacteria had been shown to activate the immune system. And I'll describe that process. The organism is called Bacteroides fragilis. It's found in something close to 25 to 50% of the human population. There's a lot of variability, as I mentioned, but it's found in close to half of the people in this room carry this organism in their intestines. And it's very unique in many ways. I won't describe all the unique processes, but it produces this one molecule called polysaccharide A, or PSA. It produces many molecules, but this is one of the molecules it produces. This organism is the only molecule that produces this polysaccharide, which is a long sugar that coats the surface of the bacteria. This is the bacteria. It's actually been stained for PSA, so that yellow color shows that this bacteria is producing this molecule. And what we discovered is that this molecule had very, very profound, very, very powerful effects on the immune system. And so I mentioned inflammatory bowel disease in humans. You can model many human diseases in mice, as I mentioned as well. And so what we did is we set up a series of tests where we looked at multiple different models of inflammatory bowel disease in animals. You can induce disease that looks like inflammatory bowel disease in a mouse, and then you can ask, does a certain therapy work in that context? And so this is an animal model of inflammatory bowel disease. It's one of the more widely used models in research, where the animals show a loss in weight, a very, very rapid loss in weight, similar to a person who has a flare-up in inflammatory bowel disease. So for those of you who know, in IBD, the disease comes and goes. And when the person has a disease, it's very severe. Oftentimes they miss weeks of work or weeks of school, and they lose up to 20–25% of their body weight, which is very, very drastic. And that's what happens in the mice. So these are animals, you're looking at weight over time, and these are animals that were induced for disease. And you can see within 2 days, they lose 15% of their body weight. Right? Once again, a very, very severe illness. These are animals that were induced for disease, but they were fed PSA. So all we did was we put PSA into their food. Right? And you can see they don't lose nearly as much weight as the animals that were induced for disease but were not treated. So it's protecting from weight loss. Even more, probably more important, is if you look at the tissue itself. So now this is an image of the colon of the animal that was induced for IBD, induced for inflammatory bowel disease. This is a healthy control mouse and its intestine. You can see how much the animal that has colitis, that has IBD, you can see how much its tissue has become thickened. Right? This is a sign of disease. The tissue is bloody. There's immune cells that have infiltrated into the tissue and have caused this syndrome, this symptom. Once again, when you look either by biopsy or by colonoscopy at a patient with Crohn's disease or ulcerative colitis, this is what you see. And in these same animals that were induced for colitis, if we treat them with PSA orally, we just give this, once again, just orally to the animals, you can see that their intestines look very, very normal. So as I mentioned, we can give the pure molecule, this PSA, or we can give the bacteria. So we can give Bacterius fragilis, this organism I told you about that produces PSA, to the animals, and it protects them from this disease as well. And not only does it protect, but we can also set up models where first the animals are induced for disease. First they get sick, and then we give the bacteria or the PSA, and then we can treat the disease, which is what you want in a therapy. Right? It's very hard to predict who's going to get sick. We treat people after diagnosis. And so there are, once again, many reasons why this occurs, and I'm going to go over this just very briefly, is that this molecule, PSA, that I'm telling you about, has very, very profound effects on the immune system. It in fact takes over the immune system of the animal and controls and regulates the immune system, and that's how it has these effects. And so, there's many facets to the immune system. I'm going to talk about just one aspect, and that's T cells. So T cells are immune cells in our body. They're white blood cells that help fight off infections. I'm sure many of you have heard of them. There are different kinds of T cells. I won't go into the detail, but all three of these T cells I'm showing you in blue are cells that are pro-inflammatory. They mount an inflammatory response. And if you can read, they protect us against intracellular pathogens, such as viruses or bacteria, or extracellular pathogens, such as bacteria or fungi. Right? This is our arsenal, our weapons against infection. But if the immune system isn't controlled, if the immune system becomes activated and remains activated for a long period of time, it will cause damage to the host, to the tissue. So once again, if you go back to the analogy of if you have a cut, if the microbe isn't cleared and the immune system keeps going, it's going to cause so much tissue damage that you'll ultimately get necrosis and you'll ultimately get a severe damage to the tissue itself. The immune system has to be controlled. And the way it's controlled is by another subset of T cells called regulatory T cells. Okay? And in fact, what we've learned over the years, and these regulatory T cells are anti-inflammatory, they suppress inflammation. And so what we've learned over the years about PSA is that it actually induces the activity of regulatory T cells. It activates these protective cells that then prevent the pro-inflammatory cells from becoming activated and from causing tissue damage. Right? And so if you think about what an infectious agent, if you now think about infections and what infectious agents do, is normally what an infectious bacteria will do is it will kill the immune cell, because the immune cell is trying to kill it, so it will kill the immune cell. But this is a friendly bacteria, this is a beneficial bacteria. So its molecules actually help the immune system, actually improve the immune function. And once again, that's the paradigm shift in terms of thinking about bacteria, the way we've all thought about them for many years, and this new class of organisms that are beneficial. And so I'm going to skip this next slide. It goes into a lot of detail about the mechanism of PSA, and I just want to always show this slide, and I'm not going to describe it. But I want to show this slide because this represents almost 15 years of work in one slide to identify the cell types, the receptors, the molecules that are induced, the location of these cells, and how they function. And so I think that for all the students in the laboratory, it's an injustice to talk about this work without showing, you know, the fruits of their discoveries. But I think for the sake of time, I'm not going to go into too much detail. But I do want to reinforce this notion of this balance between good and bad bacteria. Right? We have these organisms, many hundreds of them, and one way of looking at health, maybe perhaps an entirely new way of looking at health, is that we're healthy when our bacteria are in balance. When you have the beneficial organisms and the harmful organisms in a healthy ratio, then there isn't immune response, there isn't inflammation, right? And we're healthy. But what happens when the microbial system is imbalanced is that this leads to health, or this leads to disease. So if there's either an increase in these harmful organisms, right, or a decrease in the beneficial organisms, as I mentioned, either one of those events can tip the balance towards inflammation. So this says inflammation, it's synonymous with disease. Right? So once again, if you remember one thing, just remember this concept, is that there's no new bacteria, it's just the changes in the ratios of bacteria that were in our gut the entire time, and that can predict disease. And in fact, I'll talk about various different diseases that are induced by this mechanism.
Is it ever balanced the other way?
To our knowledge, it doesn't, because every disease where the microbiome has been involved are chronic disorders. They're not disorders that resolve themselves, right? And that's what we hope to do, is to intervene in that imbalance and to repair that ratio. That's the Holy Grail, and this is what we work hard to achieve. So the question was, one of the questions were, what changes the microbiome, right? And clearly, as I mentioned, the genetics of the individual are important. I won't discount the role of the host. It's not that the bacteria are controlling everything. Some people have a certain genetic predisposition that makes them more sensitive to these changes. Other people have genetics that make them more resilient, right? But genetics alone doesn't explain inflammatory bowel disease. It doesn't explain autoimmune diseases. But we do have to think about the genetics of the host. Yes.
I still don't understand why can't you tip the balance the other way by reducing the harmful ones or enhancing the beneficial ones?
Yes. The example I showed you is enhancing the beneficial ones, right? So if you know what organism is missing, right? So Aspergillus or PSA, you can augment or you can supplement that person and enhance the beneficial ones, right? There is no strategy to just selectively remove the harmful ones. It doesn't exist. Many people have tried. We have a project we're trying as well. We know in inflammatory bowel disease, we know which organisms are inducing inflammation. But how do you go and target one species out of hundreds and hundreds of species? So antibiotics don't do this, right? Because antibiotics kill many, many different kinds of bacteria. That's why antibiotics generally are not used to treat these types of diseases. There is no, let's say, call it a smart missile, right? Or a way to identify which organism you want to kill and selectively target that organism. But if someone were to come up with that strategy, it would be I predict it would be beneficial in these systems.
What about this new announcement of the fact that they're going to end up controlling this antibiotic-resistant bacteria? They'll be able to really attack them and control them or eliminate them. Could that be the targeted way to address that?
No, so all of those antibiotics they still kill many different kinds of bacteria. even the good ones. That's the problem. That's the problem with antibiotics. So the notion of overcoming resistance is different than what I'm talking about. So just briefly is, there are many bacteria that are resistant to antibiotics, so you have to develop new antibiotics because the old ones don't work anymore, right? But all of the antibiotics will target many different classes of organisms. And in fact, what people speculate, and in fact it's on here right, it's on here right there, right? Is what people speculate is that overuse of antibiotics has caused these issues, right? Because the antibiotics are killing the good bacteria, right? But as we mentioned, once again, I won't stress genetics any further, but diet is a major driver of the composition of bacteria. Different bacteria eat different foods, so that affects them. Of course, we already talked about how we're born. We're born in very clean, sterile environments. We're not colonized with as many bacteria as we were hundreds of thousands of years ago. We're not colonized with as many bacteria as people in third world developing countries are, right? So in this country, we live very clean, we live very hygienic, and in fact, that may be a risk factor for disease. And of course, we talked about antibiotics, and in fact, even vaccines have been implicated in this. So if you now think about all these different variables, they can each cause dysbiosis. Once again, dysbiosis is this change in the balance between good and bad bacteria. And as I showed you, depending on which organisms you have, you can either induce a pro-inflammatory response. Remember, these were the blue T cells that induce disease, and these were the red regulatory suppressive cells that protect against disease. Right? So it's a really humbling thought if you consider this, is that our immune system is being controlled by the bacteria in our intestines. And you can have a stronger immune system if you have the right bacteria, and a weaker immune system if you don't have the right bacteria. So our genes, our genome doesn't control our health, not entirely, but these bacteria are contributing to our health. And so along lines of some of the questions already, I think that
is that being approved, tested?
Is
that theory that approved, that tested, that our genome and the bacteria's is weighs heavier?
Yeah, so the studies I showed you already testify to this, because we're not changing the genotype of these animals, we're just changing their gut bacteria, right? So that tells you genetics obviously builds the organism, but it's the microbes that affect the function of the organism.
But
right?
still,
and the converse has also been true, right? Is that when people in mouse models have made slight changes to the genome, the genotype of the mouse, you can see effects on their gut bacteria. So the two are linked to each other, right? The genotype affects the microbiota, and the microbiota affects the function of the genes and their gene products. Right? And so,
how about the stress factor?
How about
Stress factor, emotion, negative emotions.
Stress? There's a set of studies that have come out, and I'm going to touch upon this just a little bit. A set of studies that have come out that if you were to stress an animal, just induce by multiple different mechanisms stress, it changes their microbiome. So stress is believed to be a component that affects the microbiome. Now, what we still don't know is how those changes will affect disease. In other words, if a person is stressed, right, their microbiome changes. Do those changes result in some negative consequence? And that's where the field is right now. And so I think all of us are familiar with the notion that if you're stressed, then you're more likely to get sick. You're more likely to have an infection, right? Your immune system is weakened. People are asking, is that through the microbiome? Right? So stress is important. And in fact, I was not negligent, and I even included stress in this diagram. And so I want to emphasize this point again. Is don't get me wrong. I think that I studied microbial pathogenesis, infectious disease. Many microbiologists are studying infectious disease. This is a very, very worthwhile area of research. We need to understand how bacteria or viruses cause disease so we can inhibit that process, right? And we benefit from this, we being people in developed societies. If you look over the past 50 years, and this is obviously an outdated slide, and this is data from the US and from Europe, there's a dramatic decrease in infectious agents, right? Which makes sense, right? I mean, we don't worry about general tuberculosis in this country, right? But what has led to this? It's antibiotic use, hygiene, vaccination, all the things that I told you affect the microbiome. At the same time, where infectious diseases have gone down, autoimmune and allergic diseases have increased in Western societies, in more developed countries. And so I told you about inflammatory bowel disease. Here's Crohn's disease. Multiple sclerosis, which is inflammation in the brain, which causes paralysis, is increasing in the United States, in Europe, and in Japan. Type 1 diabetes and asthma are very rapidly increasing in Western societies. I think with many people in this room, when you were growing up, I was growing up, we didn't think about food allergies too much. We all ate peanuts and drank milk and ate eggs. I don't know if they serve peanuts in this school, but many schools don't serve peanuts anymore, right?
Many people are allergic to peanuts.
They are, and to many other foods. And so this change has happened in 1 or 2 generations. And so I think that's a very strong argument that genetics is not the reason why there's such an increase in allergies, right? Something in the environment is changing, and we think it's got bacteria.
What's your opinion about vaccinations? A lot of parents don't want to vaccinate their children.
Yeah, so I'll actually talk about autism because autism is the one indication where vaccinations have been linked. And to our knowledge, there's no direct evidence that vaccinations cause autism. We do know vaccinations change the microbiome, change gut bacteria, right? So they do. And they do this through a very simple mechanism. Is when you're vaccinated, the vaccine itself is just usually a protein or an attenuated virus, like a weakened virus or weakened bacteria. But what comes along in that needle with the vaccine is an adjuvant, something to activate the immune system, right? This is standard in all vaccines. And when you activate the immune system, it changes the microbiome, right? Through some of the mechanisms I talked about, right? So, I'll answer the question now is, I have a 5-month-old. He's vaccinated. He will be vaccinated. He will stay on a regular schedule of vaccinations, right? There's the evidence to link vaccinations to disease is very, very minimal at this point, right? And that's just epidemiologic across many, many different societies. And I know that's a common thing people worry about. All right, so in the next few minutes or the next half of the talk, I want to discuss some of our work that doesn't deal with the immune system, but deals with the nervous system. And so about 6 years ago, we decided as a laboratory, or I decided as a direction for our research, is to think beyond the immune system. So as I described to you, when we first started working on immune diseases, we did so because of the relationship that we all believe between microbes and the immune system, right? And you saw this evidence in inflammatory bowel disease. I can tell you we also have a therapy for multiple sclerosis we've discovered. We have a therapy for sepsis, which is a massive inflammatory response in the body we've discovered. We have a company, that's one of the companies I'll describe, our first company, that's developing PSA for autoimmune diseases for inflammatory diseases, right? Diseases mediated by the immune system. But in thinking about how the immune system functions, how the nervous system functions, it became clear that there are many, many similarities. And I won't go into the details, a lot of it is detailed biology, but just go home knowing your immune system and your nervous system are very, very similar to each other. So we said, if these bacteria are affecting the immune system so profoundly, could they also affect the nervous system, right? And so we started working on autism, and I'll describe that data. And we now have a program working in neurodegeneration. So we're working on Parkinson's disease and on Alzheimer's disease. And we have some very, very promising results, and I won't share those today. But I'll talk to you about the work we've done in autism. These are classical neurological diseases, widely believed to be diseases of the brain. And hopefully I can convince you that there's more to these disorders than just what's going on in the central nervous system of the brain. Okay, so a little bit of background about autism or autism spectrum disorder. So ASD, autism spectrum disorder, is defined by 3 core behaviors. Children with autism, some of them have intellectual disabilities, but many of them are quite normal in terms of their intellect. In fact, some of them have very, very elevated intellectual capacity. Right? The disease itself this is not mental retardation. Right? This is a disease of behavior. They act differently. Their behaviors are different. Right? And the classical symptoms of ASD are impairments in social interaction. This says language and communication, and repetitive stereotype behavior. These children with autism, they don't interact with other children, right? They don't verbalize, they don't speak, right? In some cases, they never will learn to talk, right? And they also have these particular behaviors where they, once they find a task or some sort of an activity they like, they do it over and over and over again, okay? These are the hallmark cases of autism. But autism also has a spectrum of other, we call them comorbidities or symptoms associated with autism. And there's many of them, but anxiety is one. Anxiety is quite prevalent in children with autism. But I want to draw your attention to immune dysfunction and to gastrointestinal symptoms. So all the latest studies show that children with autism have elevated immune responses. They essentially have a chronic inflammatory condition. You can look in their blood, you can look in their gut, in their intestine, you can look in their brain, and you see markers of elevated immune response, right? Once again, no one had looked at or thought about autism having an immune component until about 2 or 3 years ago. Children with autism also have very severe gastrointestinal symptoms. In many cases, they have constipation, abdominal cramping, even bloody stool. And so you're thinking about this now. It's autism studied as a neurological disorder. There are symptoms, very profound symptoms, which are both immune or gastrointestinal. Right? And this, once again, caught our eye, is that based on the latest studies, up to 90% of children with autism have gastrointestinal or GI deficits, or intestinal deficits. The other thing that was really interesting to us is the same as I showed you for autoimmune or allergic diseases, the rates of autism are increasing, and they're increasing very, very dramatically. And so just in the past several years, and once again, it's hard to see, but this is 1975 to 2008, and all the way up to 2012. The prevalence and the rate of diagnosis has increased dramatically. About 50% of this could be explained by the fact that there's increased awareness, right, and increased access to medical care. Right? The other 50% is completely unexplained. Why is this disease increasing in number? And in fact, this is a study from the Center for Disease Control, or CDC, which showed in 2012, 1 in 88 births result in a diagnosis of autism. In 2014, that number is 1 in 68. So this is an epidemic. This is a problem that we all deal with. I know several children, very close people in my life with autism. And so this also indicated to me that there was an environmental component. So putting the GI, the gastrointestinal symptoms, along with these potentially environmental components, we started studying autism.
And you found any difference after treating them?
Yes, and I'm going to show you. I'm going to show you that data. But once again, it's all mouse work, and I'll talk about what's been done in humans as I present the mouse work. And so I'm going to briefly go over the animal model because I think there's a lot of detail here. But I want to enforce that animals are a good place to start to study human disease. But of course, a mouse is not a human. You know, it has many features that are similar, but many that are different. And so, as biologists, you know, you can't just take an idea and put it into humans. It's unethical, right? And so you have to start somewhere. And once again, you know, mouse studies are predominantly the way that the biological research starts. So we set up a mouse model where we induced symptoms of autism based on an environmental risk factor. And that environmental risk factor is maternal infection. There are other environmental factors. And of course, there are genetic causes to autism, right? But we focus on, and for the reasons I told you, we focus on environment. And so there were many studies. Initially, the 1964 rubella outbreak in this country, but then large epidemiologic studies, both in Denmark and in Sweden, which showed that children who were born to pregnant mothers who had a very severe infection during pregnancy are at an incredibly high risk of autism. This is not a simple cold. Right? It's not a simple sore throat during pregnancy. It's a severe infection that also involves a severe fever, prolonged fever, high fever for over a week. That's the risk factor. We modeled this in mice, and essentially we gave mice influenza or the flu. So we took mice that were pregnant, we gave them the flu, right? So that's shown here. We call this model, we named the model the maternal immune activation model, because what were we doing with the infection? We're activating the immune system of this mouse. And what we've shown over the years is that in this mouse model, the animals have all of the behavioral symptoms of autism and all of the neurological pathologies of autism. And all we're doing is getting healthy, otherwise wild-type animals influenza, the flu during pregnancy. Once again, it's a risk factor shown in human studies. And so, as I mentioned, there are the 3 core behaviors of autism, and we see that we see all of these in our animal models: impaired social interaction, decreased communication, repetitive behavior, as well as anxiety and other disorders. And when we look in the brains of these animals, when we dissect the animals and look in the brain, we see all of these hallmark pathologies which have been shown upon autopsy of human brains, children with autism. Okay? So we think that this is a fundamental reproduction of the disease in animals, because we see both the symptoms and the neuropathologies. So we use this model to test now the role of gut bacteria, the role of the microbiota in the disease progress. So the first thing we did is we asked a very simple question, and we said, is the gut bacteria of animals with autism different than that of control healthy mice, right? Just like I showed you with the human data with inflammatory bowel disease, we want to know, are there differences? And so the saline in green, saline is just salt water. This is an injection of just salt water, so that's our control, right? These are healthy animals in green. Each of these symbols represents the microbiota, the gut bacteria of a mouse. And we use computational tools and algorithms to assign a numerical value to the different ratios of bacteria, right? But suffice it to say, each point, each dot, represents, let's say, the picture or the profile of the microbiome. All of the mice in green that are healthy are in this region over here. All of the mice that have symptoms of autism are shown in blue. And you see that all their microbiomes are different. Their gut bacteria are different. They're different from each other, and they're different from the healthy animals. So then we ask, can we give a bacteria that affects the immune system, that then suppresses the immune system, and can we restore this microbiome defect, right? So we went back to our old friend, Bacterius vagos, which we showed is effective in autoimmune or inflammatory models, and we fed them to these animals that had features of autism. And that's what is shown here. So these are the autistic animals that were given Bacterius vagos, and you can see all their microbiomes are now much more similar to the healthy animals than they are to the animals with autism. So it's like there are hundreds of bacteria in these mice, but giving this one bacteria restores this balance. And this is the question of course asked, right? So that shows you the power of these particular organisms. And then now just to respond to that, you can't buy this organism at Whole Foods, right? Mm-hmm. This is not a commercially available organism. In fact, this is the asset for our second company is to develop this organism, this live therapy for autism.
How about the difference between the animal gut and the human gut, bacteria-wise?
Very, very similar. So I actually have slides on that. Very similar in terms of the overall composition. Of course, individual species will be different, but of course, individual species between us are different as well. But the mouse microbiome is very, very similar to that.
Why we cannot buy from the store? Because it's not safe for us to use it, or it's not developed yet?
It's not developed yet. And so the FDA, the Food and Drug Administration, as of 2010, doesn't allow anyone to do a clinical trial with a bacteria until you get approval. Right? Every organism before 2010 that was sold as a probiotic no longer, it does not require this test, because it's already been taken by enough, and the rationale by the FDA is it's already been taken by enough people, enough humans have taken this bacteria that we believe it's safe, so you don't have to do a safety trial. But in 2010, they changed the regulations, And any new organism that has to be introduced, you can't just take an organism and put it on the shelf. It has to go through a clinical trial. Right? And that's what we're doing now with the company, is we're raising money to do a clinical trial, both for safety as well as for efficacy in autism. Right? And so if we had done this work 10 years ago, we could just give out the organism. But now, by law, I can't do it.
Is that also means that, I know there are millions of them, but each and any micro organisms are specifically to cure specific, let's say, diseases?
Yes.
Could you repeat the question?
Yeah, so the question is, are specific microbes specific for certain indications, certain diseases, and not for others? And the answer is yes, because after we tried Bacillus fragilis in this model, we tried over 10 other bacteria, and they did not have any benefits whatsoever. Right? So there's something special about this organism that isn't found in other organisms that live in the healthy human gut. Right? And we don't know what it is. We don't know what's unique about this bacteria that's different from other bacteria. We just know that the bacteria works. Right? So it's not that you can just take any bacteria. Right? And so this is one of the things that I tell all my students is, you can't just look under the microscope because all the bacteria look the same. But if you look at their DNA and their function, they're all very, very different from each other. Right?
Excuse me, are you going to talk about the familiar Mediterranean disease?
I am not going to talk about familial Mediterranean disease known as the Armenian disease.
Yes.
Right? But there is, and I will show her a picture, there's a researcher in Hayastan. Her name is Jana Katotsan, and she's been studying the microbiome of FNF patients, and she's shown that their microbiomes are different. Whether or not the gut bacteria are causing this or potential therapy, we don't know. But people in Hayastan and at the NIH, at National Institutes of Health, in Bethesda and Washington, D.C. are also studying. My laboratory does not study. Okay, I'll show you a brief cartoon. This is supposed to be amusing. Let's see how it goes. To describe some of the behaviors of mice in these various settings. So first I mentioned anxiety. This is an anxious mouse. Okay, so how do you measure anxiety in an animal? The cartoons get better, trust me. So how do you measure anxiety in an animal? Right? There are many ways of doing this. We use multiple different behavioral tests, and the one I'll describe is called the open field exploration test. Right? It's a very straightforward measure. What we do, we take an animal, we take a mouse, and we put it into a cage that it's never seen before. So animals are very good at scent. They smell things, so you want to put it in a fresh cage so it has no biases, no fears whatsoever. And mice have an intrinsic fear about them. Mice are anxious by nature. If you think about this, the mouse is prey to many predators, right? So by nature, they have this instinctual anxiety. Right? But at the same time, they're also curious because they have to go exploring for food. And so the way you measure anxiety in these animals is you put them in this cage and with a camera, you measure how long they spend at the edges, right, which is a safe place for them, versus how much time they spend in the center of the arena, which shows a lack of anxiety. Because remember, the animals are fearful, but they also have to go out looking for food, right? So we have a camera-mounted system. We use computational tools to analyze how often the animal goes into the center and how long it stays in the center. The more it enters the center, the less anxious it is. The more time it spends in the center, the less anxious it is. So here are the results, and all these results were published just last year, is if you look at center entries, how often it goes into the center, or center duration, how long it spends in the center, you see that the control animals, this is in black bars, the saline animals, they enter the center and they spend more time in the center compared to the autistic animals here in the white. Right? So the animals that were induced for autism have this anxiety trait, right? Where they exhibit this fear, this heightened sense of fear. And if we were to take the autistic animals and give them Bacillus fragilis, this probiotic, you can see that the deficit is restored. Their disease is now normalized. And we're giving this to adult animals. We can take animals that already exhibit anxiety traits, just feed them the bacteria, and their anxiety goes away. Okay? And so we've done this test. We've done several other tests, once again, as well, and we see the same results. That in anxiety tests, the bacterium is able to cure their anxiety. So I mentioned compulsive behavior as a phenotype. And here what we use is a marble bearing test, and you'll see this animal bury the marble. This is a normal behavior for a mouse. But a compulsive animal has this behavior, but it's quite extreme. It buries many, many, many different marbles. Right? So, you have to understand the perspective of an animal. Mice are playful creatures. I've worked with many of them over the years. And if you put a novel object or a toy, they play with it. Right? If you put a marble into the cage, they'll bury that marble. Right? But they'll eventually get bored and move on to a different task. Right? So to measure this compulsive, this repetitive behavior, all we do is we take a cage of bedding, so you keep these animals in a cage, and we just line up marbles on top of the bedding. And the bedding is just wood chips that the animals sit on. And in a given amount of time, we ask how many marbles did they bury, right? And even the healthy mice will bury some marbles. Once again, this is an activity for them. But an animal that has a repetitive behavior, that has stereotype behavior, once it buries 1 or 2 marbles, it feels compelled to bury the next one, and the next one. And in fact, this is what we see. So these are the control animals, and in a 10-minute period, they bury 30% of the marbles they see, whereas an animal with autism buries about 45% of the animals they see. There's a significant increase. Right? So now you see an increase or an addition of phenotype. And once again, in the animals that were given the probiotic, we see that their repetitive stereotype behavior goes down. So we can actually treat this core behavior as well. Oh. Never go. Give me 1 second. Okay, I don't know why that happened. All right, so finally, as I mentioned, children with autism don't verbalize, they don't communicate, right? So we can measure communication in mice. And so mice communicate at an ultrasonic wavelength. And so they don't communicate with a wavelength that's audible to us. We can't hear a mouse. So when a mouse squeals, if you listen, you've ever had a mouse in the house, if it squeals, that's not how they talk to each other. They talk at an ultrasonic wavelength. And so we have a microphone, special microphone, that we can use to eavesdrop on mice and to listen to them talk. And I'll see if this works, but you can hear it. This is the sound of a mouse talking.
In Armenia?
Stop,
Sounds like my wife.
And so we're actually trying to decipher in a separate project what they're saying to each other. There's syllables, there's actually sentences, right? So we're working with a computer programmer to understand this. So once again, we use this as a measure to understand the vocalization or the communication between animals. And as you can tell, animals with autistic features, they call less, they make less sounds, and the duration of their calls, the length of their calls are reduced. And once again, the probiotic restores this classical feature of autism.
How do you induce the probiotics to the mice?
How do we produce it?
No, induce.
How do you give it?
Oral.
Orally.
Oral.
We just give it orally. We give it 1 time to the animal and calm it.
Interesting question. Are these mice of both sexes or just one sex? Because this sounds more like female mice.
It's actually male mice. Right? So there are two ways of doing it, and I'm presenting some of the data that we gathered in the laboratory. There are two ways of measuring communication in an animal. You can measure a pup, an infant mouse calling its mother. So when they're nursing, if you separate them, they call, right? But this paradigm is a courtship paradigm. So you put a male animal with a male animal that's you're testing with a healthy female animal, and essentially, are they engaging each other? Is the male engaging the female? That's what you're testing.
Yeah, yeah.
Sure.
Speaking of males and females, there are more male autistic kids.
4-to-1 ratio of males to females. Yes, we see that in the mice as well.
That means because males organisms are, I don't want to say weak, but more perceptive to that disease?
No one knows. So no one knows what causes the sexual dimorphism in fact, in any disease, let alone something as complex as autism. So if you think about other disorders, there are, like lupus it's an autoimmune disease, more common in females than in males. No one knows why, right? In autism, no one knows why males get the disease more than females. This is speculation. What we do know is that gut bacteria in males and females are different, right? So gender does control the microbiome as well. Is that the reason why there's a sex bias? We don't know. But the fact that we see in the animals makes us feel like we are reproducing human disease.
In this respect, have tests and experiments been done with twins?
Yes, they have. Yes, they have. And both monozygotic twins, right, and dizygotic twins, right? And in fact, in monozygotic twins, the concordance rate, meaning how often do both twins have the disease in autism, is 70%. Okay? That tells you, even though the genetics are identical, that they're not always going to get the disease, that there's an environmental factor. Okay? There's other complexities to this. Do they share the same placenta? There are differences there as well. So a lot of these types of association studies have been done.
So inheritance has some kind of influence on
Genetics, yeah.
inheritance does actually come into play.
Autism is not a heritable disorder.
It isn't.
It isn't, right? It does run in families, but it runs in the same generation, right? Not through generations. But this is, and that's the data, right? But this is a complex issue because most people with autism don't reproduce. And so you don't really know whether or not they're more prone to have children with autism, right? But the parents generally don't have autism, right? And so if it's inheritance, and I've actually written an article about this, if there is a heritable component, and there certainly may be, right? Remember, how do we get our microbiome? We inherit it from our mothers, right? So not only do you inherit genes from both parents, but you inherit bacteria from your mother. So you can horizontally transfer information, in this case bacteria, to your offspring, right? So there may be a heritable component, but the heritable component may not be genetics. Right? That's speculation. Okay, so I want to talk about the therapies, right? And that's obviously probiotic foods. And so we feed the animals the probiotic.
It doesn't get destroyed on the gastrointestinal tract?
Yeah, we buffer it. We buffer the organisms, so we encapsulate them in sugar coating that allows them to penetrate through the stomach and get into the colon. And all this says is the data I've already shown you, is that animals that are treated are essentially happy and don't have the symptoms of autism. Okay, in the sake of time, I'm going to skip this slide, but I'm going to tell you the actual phenotype that I already described, is that mice and humans with autism have GI deficits. And the main phenotype that we see both in mice and humans is leakiness of the gut, right? We see permeability of the intestine. And what that means, essentially, is if you think about the role that our intestine plays, right? Our intestines have to keep certain things out, like toxins, but have to let other things in, like nutrients, right? So the barrier is very important, and this is one of the main functions of the intestine, is to decide what to allow in and what to keep out, and obviously in terms of when we excrete byproducts from ourselves, what to let through the intestine, to go out and not to come back in. The phenotype that's been shown in humans over and over, in addition to the symptoms of constipation I told you, is that they have leakiness of the gut. And what this means is that the barrier, the wall of the intestine has holes in it, essentially. We tested this in animals, and I won't show you the data, but we see the exact same phenotype in these mice. We do this, we take a sugar molecule that is too large to pass through a healthy gut, and we feed it to the animals, and then we look in the blood for that sugar. In the healthy animals, we don't see that sugar because it's of a large size. In the mice with the autism, we see the sugar seep into the gut or leak into the gut, right? So there's some barrier defect in the intestine of these animals. And so what we ask is a I think a very sophisticated question is, if the gut itself is permeable, if there's now a deficit in the barrier, do things get across the intestine that then cause these behavioral deficits, right? And I think this is a very important point. So as I mentioned, as many of you already perhaps believe, is that autism is a disorder of the brain. This is what classically every neurologist, every behavioral biologist believes, right? But we wanted to explore the hypothesis, and it's a very revolutionary idea, that perhaps autism starts in the gut and not in the brain. And that it affects the brain because things leak out from the intestine and get into the brain and cause these deficits. Okay? I know, once again, very controversial, but we tested this. And the way we did this...
You had a gut feeling that makes...
I had a gut feeling that the neuroscientists were wrong. Right?
What's the history of this kind of diseases? Ancient people had this kind of diseases, or their diet and lifestyle was different,
no one knows.
and they didn't have it.
Yeah, no one knows, right?
Because the disease you're saying about autism and Armenian Mediterranean disease have almost the same symptoms in the intestine.
Mm-hmm.
So it is very interesting to find a correlation or difference. So it additionally proves the things which you're talking about. If the bacteria are doing this thing, find out if the bacteria are the only reason, or what is the difference between them and reaction with the body and this kind of, it helps to
compare among diseases, right? Is the question. So we're doing that in the models that we work with, and that's IBD and autism. Both obviously have a both have a gastrointestinal component. So we want to know what's the same and what's different. There's a lot that's different, I can tell you that, right? On many levels. So we can pull out individual cells, molecules from the intestine. The profile of the immune system, the nervous system in the gut between IBD models and autism models are very different. And so I agree with you, is if you find something that's similar, then you might get to the heart of what's causing it, but we still have not yet done that. With something like FMF, I think that's, once again, we don't study it, I'm not an expert, I think that's even further removed, right? But in terms of the other question about what happened in ancient societies, of course no one knows, right? Because autism diagnosis requires a certain test, a battery of tests that behavior biologists, have been doing for only about 20 or 30 years. They've been doing it since the 1950s, but it's really only been widespread for 20 or 30 years. So we just don't know.
Okay. A question. So if that theory has merit, right, that there's permeability in the intestines of autistic children, isn't that easy to detect on the children themselves? I mean, there are ways to find out, right?
Yes, and so as I mentioned, many, I can't quote the number, but it's over 50%, it could be 75% or higher of autistic children that have been tested have leaky gut. So they have this symptom. Right? And so one thing, we wouldn't, so we do see, and you bring up a good point, because we do see symptoms in animals that we don't find in humans, and we don't follow those symptoms. Right? There are certain things that, because an animal, once again, a mouse is different than a human, right? But the reason why we're studying this is because when we looked, and we found this, and we read in the literature, we saw what we were seeing in the mice was exactly what was reported in humans. Right? So we feel like that is an important feature of the disease. And so I want to just end in the last couple of minutes with what is connecting the gut and the brain, right? So if you've been following this, and if you believe any of the data that I presented to you, I'm talking about reactions that occur in the gut. But we know there's neuropathology in autism. We know there's behavioral deficits in autism, and that's widely believed to be controlled by the brain. So how is the gut communicating to the brain? And so what we did is we looked in the serum of these mice, both the control animals as well as the animals that have symptoms of autism. And we asked, can we find small molecules in the serum that are selectively upregulated or increased in the autistic subjects, autistic mice? So if you think about this, there are many chemicals in our intestine, both from food and those produced by bacteria. If the gut was leaky, you should see more of those in the serum, in the blood of the autistic animals, because more leakiness, right? And in fact, that's what we saw. So we identified one particular molecule called 4-ethylphenylsulfate. It had only been described in 2010. So before that, I joked that God created this molecule in 2010. But we found it in our screen. And what we discovered was that healthy, normal mice have low levels of this compound. But when we looked in autistic animals, they had high levels. And so this is a cartoon, but we have exact micromolar concentrations of this molecule. And so this told us that perhaps maybe there was some merit to this leakiness, and maybe it was causing a defect. So then we asked, where is 4-ethylphenylsulfate coming from? Is it coming from food? Is it coming from the environment? It's only produced by bacteria. Our bodies don't make 4-ethylphenylsulfate. The mouse doesn't make 4-ethylphenylsulfate. This goes back to what the genome of the animal produces, what the microbiome produces. The gut bacteria make this molecule. And in fact, those gut bacteria that are increased in the autistic animals are the ones that are producing 4-ethylphenylsulfate. So I didn't go into all the details of which, because remember there's hundreds of species. Some go up, some go down in autism. The ones that go up in autism are the ones that are producing 4-ethylphenylsulfate. Okay? So this made us think perhaps this molecule was inducing disease, was causing behavioral diseases. And so the first experiment we did was to then ask, if we treat these animals with Bacteroides fragilis, does the 4-ethylphenylsulfate go down? And the answer is yes. Right? So if we take these autistic animals, give them Bacteroides fragilis, which repairs their gut, which prevents the leakiness, now the serum levels of this molecule go down. Right? But here was the proof of concept experiment. If this molecule is increased in autistic animals, then one can speculate that if you just take a healthy, normal animal and just inject it with this one compound, that the animal should have behavioral deficits. Right? If this molecule was causing disease because it's getting out of the gut, right, then you can give it to a healthy animal and induce autism. So first we did an experiment where we orally fed 4-ethylphenylsulfate to a healthy mouse, which has no leaky gut. We looked at its behavior, the behavior was normal. When we injected that molecule, 4-ethylphenylsulfate, into the mouse, so now we bypass the gut, we're essentially reproducing the leaky gut, this is what we see. So once again, this is injection of 4-ethylphenylsulfate into healthy mice. We do the open field test. You can see animals that received 4-ethylphenylsulfate enter the center less, or they spend less time in the center than the control animals, which just received saline injection. Okay? Remember, this is a measure for anxiety. The less time they spend in the center, the more anxious the animals are. So what have we shown here? And so this just shows that they're traveling the same distance, which is a control. It's an important control. But what are we showing here? Is that you can take a molecule produced by bacteria, right? inject it into a healthy animal, and cause symptoms of autism. We've done this with phenyl sulfate with anxiety. We now have another molecule we've identified, indopyruvate, which causes repetitive stereotype behavior. We inject this one molecule into mice, and they bury more marbles than mice that didn't receive this compound. That molecule, indopyruvate, is also produced by bacteria. So I think you're getting the picture here, and I'll conclude with our model for, and this is, once again, very, very revolutionary, very, very controversial to say the least, is that perhaps autism, perhaps even Parkinson's and Alzheimer's, which have GI complications, for those of you who know, are diseases of the gut. They are not diseases of the brain. And if you treat the gut, you can treat the disease. Right? And what we've shown in these mouse models is that not only are there differences in the microbiome of animals that have autism, there's also this leakiness of the barrier. So what you're looking at here are the cells that form the barrier of the gut, and you see the leakiness. And if these compounds, if these chemicals get through, then they cause disease. If you treat with a probiotic, this particular probiotic, you can not only restore the balance, but you also now don't have this leakiness. The junctions, the tight junctions are formed well. The barrier of the gut functions as it should. Molecules don't get across, and the animals don't show symptoms of autism. And so what does this mean? I think that, we, myself included, we take a lot of drugs, we take a lot of chemicals, a lot of pills. This is the society we live in, is that we are tied to medication. But perhaps there are more natural ways to address or perhaps even treat disease, and that's think about how humans evolved, what microbes we evolved with, what are the changes we've made in society that have changed our composition with microbes. And can we go back? Well, I won't advocate that we go back to a time where we all have smallpox or tuberculosis. I mean, that's certainly not what science and medicine wants to achieve, right? But if there is a harm, caused by the society that we live in, perhaps the way to go back is to then give back the organisms that we're missing. The organisms that we evolved with for millions of years, but now we've lost them. So if you restore those, if you give them back, then maybe that's a therapy. And I like to show this picture that, there's an arms race, right? We take a lot of antimicrobials. We use antimicrobial hand sanitizers. They generally don't work. I can talk about that for hours. Yet our association with the world itself, this is just one small association with the world, may be the natural therapy, a natural cure for many disorders, immunologic, metabolic, neurologic. So this is the members of the laboratory. So I have a wonderful group of students and fellows and visiting professors. I won't thank them all, but I want to thank Elaine Tsao and June Round, who did most of this work. I have very, very close collaborators at Caltech, at Baylor in Texas, and Bana Jabri, who's Armenian in Chicago, who's helped us with many of the immunology work. All of this work that I showed you was done at Caltech, and we are continuing this work both with autism and with the immunologic disorders. And I want to end just with a couple of notes about the connection to Hayastan and what does this mean for our country. As Hagop described, I teach in Hayastan every year at the Molecular Biology Institute. So I don't know, if you visited Molecular Biology Institute, so this is it right here. So this is the building, and that's me. This is Haik Eskandarian. He's a fellow at the Pasteur Institute in France. This is Bana Jabri, who I just mentioned. She's a professor at the University of Chicago. She's Armenian as well. And the three of us, we go every year and we teach. We are supported and invited by Anna Boyadjan, the director of the institute. These are some of the other professors. And most importantly, the Usanovna. These are the students who come from all over Hayastan, not just Yerevan, they come from all over Hayastan, from Artsakh. Every year, about 50 students come to take this course. It's a one-week course that we teach. And I can't tell you how intelligent these students are, how dedicated they are, how Ashkada said, how devoted these students are. In fact, in many ways, I think that they far surpass the students in this country in terms of their will. What the Michot Chunin, Pites, Yev Khelatien, Pites Usumat Chunin, Spachta Kirker Gartan, Pites practical Chenkider, laboratory quantitative Chunin, DNA sequencer gets Arpanger. Arpain անդամների ինչ-որ թարմալակեցին, որ սեկվենսը կեն, որովհետև ասանք կոլզիկներով, ասանք միջոցներով պիտի հարաճանա Հայաստանը։ Իմ գոմես ես ինչ կնամ, միան ուսում կնամ տալ, քիչ մոտ բան Thanksgiving տրամա կնամ տալ, անյա կարևոր է։ Բայց ամեն կարևորն է, որ ժամանակ տաս, որ ամենից ապեր ժամանակ տանք, ինչու խոսենք այսօր, ինչու Արփայի անդամների և ամենիսն նույնպես չանք դուրանք Հայաստանի աբակային համար, որ միան տրամով միջի հարցը լուծվի, բայց մեր ժամանակով լուծվի, որ ժամանակ նիստ տանք Հայաստանին, that's why government are ինչու գերտամ Հայաստան ուտան, և նույն lecture, այս նույն lecture դվի, բայց ամեն դարի տարբեր lecture-ներ ուտանք, տարբեր նյութերի մասին կխոսենք, որ աշակերտները ամեն դարի նույն աշակերտներ կան և տարբեր աշակերտներ կան և ինչ նյութեր, որ այն դարի հարաճացած են այն նյութերը սովեցնենք իրենց։ Վգայգան ուտանք աշակերտներում, ասի մեր նվերն է իրենց, ասի հայգն է և աշակերտին վգայգան ուտա, որ ասի մեկ շաբաթ թասարանը արավ, և աշակերտները ինչպես ասի շատ ուրախ են, շատ հբարդ են, որ աս թասը կարեն, բայց ավելի հբարդ են ես, որ կրնամ ուտալ Հայաստան։ Եվ ամենամեծ նվերը, այս մեկատ նգար, որ քիչ մխնդալիկ է մեզ, ամեն դարի ինչ հյուրասիրությունը մեկատիկ է Հայաստանի մեջ ինչ-որ գիտեք, ամեն դարի մեզի նվեր կտան։ Միշտ կստեմ, որ Հայաստանը լանը, ծեր մոդը լանը ինձ ամենամեծ նվերը ասի է։ Բայց հստակ գիտեսնենք, որ կարծես այս տարի ինձ կխարտ մտնում, չեմ վսած չեմ ինչև, բայց ուսուցիչները, ուսանողները թամը կհավաքեն, ինձ միշտ նվեր կտան։ Ասմդացումով ցեցկեմ, որ մեր ամենամեծ նվերն է Հայաստանին, մեր չանկը, մեր ժամանակը, մեր ինչ որ մեր արհեստն է, ասի Հայաստանի տակ։ Շնորհակալ եմ։